primers we used in yours, Manal or Lombardi at all?
I think that's correct, and those primer
And
pairs...
so there's another family member there. It reacts with our antibodies in a Western format.
The Silverman lab designed those primers specifically to avoid contaminating MLV sequences because they were aware of that problem. So those primers were specifically designed not to detect
it's
that.
It's not a contaminating MLV when it's in a human cell.
I can show you some more slides which I had to take out where we devised an assay at NCI that will detect both kinds of viruses and in fact distinguish them in a rapid test one from the other, and would easily detect anything that's on that tree. Unless it had a big gag deletion or something like that, anything that might be infectious on that tree, this test would.
So would this?
Can
I just ask a question? Because again, not being a retrovirologist, would deep sequencing resolve these issues? Would we be able to see other unnamed retroviruses using this newer?
Yeah, but I mean, deep sequencing won't resolve issues of contamination. If the contaminant is there, deep sequencing will pick that up very nicely. It might show other things.
It won't be integrated.
It's dependent. But it's again dependent on the primers that you. Deep sequencing is based on. You have to. The way we do it now, maybe someday somebody will figure out how to do it without doing this. But the way we would have to do it now is to use PCR to start. Okay, so you could deep sequence the PCR products, and then you could find minority species in them that might let you discard things that could be potential contaminants, and then focus in and maybe find something else. It might be an approach that might be worth doing. And there are people, of course, that have devised much more broadly specific deep sequencing kinds of approaches that will detect almost any virus, and that would be I think, very effective to try to apply that kind of thing and to see what's really there in all of these.
But integration and flanking cellular sequences show it's an integrated provirus in a human infection, just like the Petros lab and just like the Sam Chow has shown.
The problem is, of course, it's been pointed out that one of Sam Chow's sequences must have come from a contaminating cell line. We don't know about the other dozen or so that he published, but at least there is some contamination there, too.
We also screened the NCI 60 cell line screen, so I don't know about widely used, but the 22RV1 cell line is not in the NCI 60 cell line screen, nor was a single cell line in that screen infected. We do the cell line, the LNCAP, in our lab every single week. We do the PCR. We don't see proteins in that. It's not a PCR contaminant. We've done everything possible to rigorously control for contamination. We don't even have cell lines in the labs where we do the blood. We've sent the blood from independent phlebotomists to different labs around the world, and they find the virus there without any manipulation or passing through our lab. There's just no evidence in our study for contamination. There's more evidence for a family of human gamma retroviruses that are involved in infecting humans and circulating in the population.
I cannot address what's going on with sequences that I don't know.
It's not sequences, it's
with
proteins.
With viruses and proteins of sequences that I don't know. If we get these sequences of these other viruses into GenBank where people can look at them, then we can continue to do some analysis.
We put
continue
the
to talk.
analysis up there.
John, you're not going to be here tomorrow. I would like to pick your brain to know, what do you see as next steps for XMRV, MLVs?
I see the next step as leaving the virus that we know as XMRV behind. I don't see how that. Now that doesn't mean there's not another gamma retrovirus that is not sufficiently cross-reactive with the primers and things that have been used to not be detected. I think I think, however, that enough evidence has been presented that there is some infectious cause here, and maybe another retrovirus. It's entirely possible that it's worth continuing to do it. I would say, however, that we are continuing to participate in studies like the blood working group studies, like and a study that's enrolling in seeing patients right now at NCI. The patients are coming in, are being collected. Patients who've have been told that they were XMRV positive by various assays, and we are going to do a very thorough workup on these patients using exactly the same sort of conditions that I mentioned, where every patient is patients and controls are brought in, treated as much as possible blindly. At least every single sample that's taken is taken using exactly the same set of reagents and tubes and so on and so forth. And those are going. You're just going to work those up through all of the assays we have available at the NCI. And the NCI, I have to say, has done a huge amount of work on this. It's taken a huge amount of resources and diverted it from other things to work on this problem ever since even before the science paper appeared. Stuart Legreese, for example, who's here, has done an enormous amount of effort into kind of shepherding these studies and coordinating them across the whole institute. And these studies are going to continue to go on for looking specifically at XMRV for MLV-related viruses and other things. And so we feel we've really, for sure, gotten to the bottom of this. The Ian Lipkin study, as far as I know, is still planning to go on, where patients.
If I can interject a second, now that we've resolved this. Now, I think the data for the recombination and the origin through the transgenic mice or through the nude mice is very convincing. I have to say that, and what is difficult for me is the next step of how it contaminated the laboratories. And you can say, well, it's in a reagent, it's in a tube, but every laboratory, including the low laboratory, uses multiple controls on every run, and they're always negative. And we have looked for he has looked for mouse genome first by a very, very sensitive nested PCR that was 100-fold more sensitive than the PCR used to detect the polytrophic viruses, and did not find any genomic DNA. Then used your first IAP assay and then your improved IAP assay exactly as you gave us the formula, and do not find any contamination. Now, you can't rule it out. You can never rule out contamination 100%. But it isn't there, and I still don't know why the negative controls in Judy's lab are negative, and why on the panels that have been done thus far, she's been able to distinguish samples from patients versus negative controls. All that be it as may, I'm not denying that there may be contamination. So I just want to put this in perspective, as Dr. Coffin has alluded to. There are two panels now that are well in process of being prepared, but have not yet been tested. The first is from NHLBI, taking patients that Judy has identified as XMRV positive, sending them to a central lab where they're being recoded or being coded, and with appropriate controls that have been confirmed by many labs as to being XMRV negative. Those samples, after the coding is done, are going to be distributed back to Judy, but she won't know what they were, to low lab and to CDC. That's a rather small panel, but to look at that particular group. The NIAID Lipkin panel, I think, will be the definitive one, because the starting point was to take patients from multiple different CFS expert centers, patients that have been very well pedigreed and followed, fit all the classic definitions. Those samples were drawn in large volume, being aliquoted and made into different fractions, and being sent as triplicate codes to the same laboratories. And if they can't break the code, then that means there probably is no association, that the published associations are not real. If they do break the code properly and don't find the negatives and do find it in the patients, then you will know it's not just contamination. And then you go from there to look at causality issues. So this is really a critical piece that's coming down the pike. Okay.
Yeah, there is in addition an NCI-sponsored study that, in fact, I think blood being drawn from patients even as we speak, just over across the street at the clinical center, which will do a very intensive workup on a relatively small number of patients and match controls. And that, as I say, is being worked up now, and the result of that will probably be available before any of these other studies.